Hi everyone, and welcome back to Diabetics for Diabetics, the go-to place for all things current diabetes research, where throwing around medical jargon isn't enough. We're not looking to piggyback off of the current medical narrative. I'm your host, Bowie. I've had type 1 diabetes for over 20 years. I'm here diving into the scientific research, looking for tips, tricks, and explanations that are going to help us, the people with diabetes, to better manage our condition. And today, the paper we're going to be looking at is the metabolic signatures of dietary iron overload in a mouse model. It was published in Cells magazine, 2018. And what this paper did was took two groups of mice, fed one a standardized diet with a typical amount of iron in it, and another group with a very large amount of iron in it. And over a 7 and 14-day period, looked at some of the downstream metabolic byproducts in each of these groups to compare what was happening at the metabolic level, the cellular level, inflammatory level of these mice on these two different diets. So let's take a look at what they found.
So here we are with our paper, Metabolic Signature of Dietary Iron Overload in a Mouse Model. So in terms of what we're looking for, the specific areas where the researchers here were looking for the effects of iron were in the blood, so specifically related to blood cells and blood tissue, or the red blood cells, the plasma, which is the fluid in the bloodstream that isn't the blood cells, and then liver tissue specifically. Now, they were paying attention to the liver because of the liver's primary role in iron regulation. In any condition where iron is present in excess or too little, the liver is going to be the first place to feel it because that's where liver is recycled, that's where liver is stored, that's where liver is repurposed in the body. So in the introduction, the paper goes through some of the basic concepts behind iron dysregulation in the body, specifically the increased production of ROS, which we are familiar with in the development of diabetes as well as affecting bioenergetics, so altering glucose metabolism and fatty acid metabolism at the same time. And another cool word that you may not be familiar with is this metabolomics, or metabolomics, which is essentially the study of metabolites within a specific organism. Now, this paper is using a new resource available to measure specific metabolites within the mice who are undergoing these different dietary interventions. And what this allows for is to see what energy systems, what regulatory systems, what excretory systems are being used as this food is being taken over a span of time. So I mentioned in the introduction, this is at 7 days and at 14 days, the researchers are able to take like a snapshot of the levels of particular energy systems, particular chemical reactions within the body, to see in real time how the body is adapting to the dietary interventions. So let's take a look at the results and some of the metabolic signature changes that were a result of this iron overload. So here in figure 1, they are graphing out the changes over time of 6 specific different metabolites that they were focusing on to paint a picture of the oxidative stress, the inflammation, the energy systems being used. Now, those 6 are glucose, malic acid, methionine sulfoxide, cysteine, 2-hydroxybutyric acid, and lactic acid. Those 6 all correlate with the energy systems and oxidative stress systems that I mentioned before. And as you can see, over time, there is a clear correlative difference with the diet and an increase or decrease in these specific metabolites. So in section B here, we'll talk about glucose in general. So this has to do with the presence of glucose in the blood, so like an increase in blood sugar, something comparable to insulin resistance or diabetes. Now, in the iron-rich diet, which is the orange color in each of these, it was clear that after those 2 weeks, those high iron mice had higher levels of glucose in their blood. The opposite was true for malic acid. Now, for those that are not familiar with malic acid is what I know from flushing my gallbladder. So malic acid is the plant compound responsible for tartness in green apples, in grapes, in wine, and it's something that helps soften bile stones. So it has a lot to do with the digestive process and the correct processing of fats. Again, this is all downstream of possible liver dysfunction. But it's just interesting to see that in these high iron diets, malic acid is decreasing, which has a lot to do with possible calcification in the liver or just overall decrease in function of the liver. Now, methionine sulfoxide, 2-hydroxybutyric acid, and cysteine, are more associated with an increased oxidative stress in the liver. So the more stress the liver is put under, the more of these metabolites are going to be produced. So as you can see, with that high iron diet, the liver is going through some stress. Now, lactic acid down here in the left-hand corner has to do with being a byproduct of proper glucose metabolism. Okay, so as part of the Krebs cycle or the citric acid cycle in the body in terms of creating energy, if you are properly metabolizing glucose, lactic acid is a natural byproduct of that process. This is noticing that with 2 weeks of high iron diet, there is a noticeable decrease in the amount of lactic acid. And this could paint a picture of poor glucose metabolism, which is mirrored in that high glucose in the blood sugar.
So essentially what you could be taking from this paper is that with that high iron diet in a 2 weeks time, it's clear to see that there are changes happening in both the degree of glucose metabolism. We've got an elevation in glucose in the blood, as well as a decrease in lactic acid, which is a byproduct of proper glucose metabolism, as well as increases in metabolites associated with oxidative stress in the liver, which means that the liver is dealing with a little bit more than it's used to.
Now, taking a closer look at figure 2, just below figure 1, they noticed that animals fed a high iron diet displayed increased urea blood levels, indicating a dysregulated urea cycle in the liver. Accordingly, the liver metabolome was characterized by decreased levels of ornithine, citrulline, and arginine. These are all amino acids and metabolites associated with the proper urea cycle within the liver. Now, we won't go into the specifics of each of those amino acids and compounds, but it is just further evidence that the liver is under a great degree of stress, and it is altering its ability to properly filter waste. Now, think about this in context of diabetes. Altered kidney health, altered liver health. These are all things surrounding the pancreas, right? If you look at the internal organs of the body, kidneys, pancreas, liver, gallbladder, they are all right there in the same area. And each one uniquely relies on the other in order to do its job. With the liver taking such a big hit with this altered iron metabolism, everything is going to be affected in some way. If you remember in figure 1, we were talking about altered glucose metabolism. So that means the beta cells of the pancreas are having to work a little bit differently, right? If different energy systems are being used, glucose isn't being used quite as regularly, something's going on with the pancreas. And when you think of urea, you think of urine, right? And when you think of urine, you think of the kidneys. So now, with the liver affected enough to alter the urea cycle, the detoxification happening in the liver, now you have to consider that maybe there's something going on with the kidneys as well, right? And there's such a close correlation with diabetic health as well as chronic kidney disease. Now, this is one of my favorite parts of this whole paper. Table 1, metabolites significantly changing in peripheral blood after 2 weeks of a high iron diet. So this is looking at the trends in all of these different metabolites coming from the blood cells of these rats fed either normal chow or high iron chow. And so just looking at this is all like, okay, if you are overloaded in iron, these are the results. Like these are the byproducts that your body is giving off. And when I first looked at this, it was like, okay, this is like just a bunch of hints as to which energy systems were being used, what waste systems were being used, and got me thinking on what are some ways that we can adjust for these. So you can see huge fourfold, threefold increases in AMP and ADP. These are all, you may be familiar with ATP in human energetics. So ATP is the ideal energy source for humans. AMP and ADP are signs of incomplete energy systems, right? So again, hammering home that altered metabolism, altered glucose metabolism, there's not a lot of ATP going on. And I'm not going to go, item by item here, but you are able to see an increase in glucose, the hydroxybutyric acid. That's an increase in an oxidative stress marker. So 2,3-hydroxybutyric acid is a byproduct of the creation of glutathione. Glutathione is an antioxidant made for the liver. You can also see, there's just so much to go through, and I don't expect anyone to go bit by bit and be like, okay, this is what this means, this is what this means, but it does start to paint pictures and trends for things that are not going as expected. You see a decrease in malic acid, right? So that's a decrease in the fatty acid metabolism, right? Malic acid is what keeps bile soft and prevents the accumulation of gallstones. There's a decrease in L-arginine. L-arginine is a major player in the NOS, the nitrogen reactive, or NRS, nitrogen reactive species. Again, something that fights inflammation. All of the byproducts of trying to create antioxidants are up. The antioxidants themselves are down. So that just shows that there's a lot of oxidative stress going on here. So just fascinating and a good list to work off of. And in the results, they go on to talk about how the liver metabolome reflects that iron is really making negative changes, or it's adapting to these high levels of oxidative stress. So you see here, on one hand, iron supplementation triggered liver metabolic remodeling to cope with oxidative stress that enhanced all of these pathways that are causing increases in oxidative stress. Now it's a matter of looking at what the paper is, the information, the data that this research is giving us. And now we have to start to think, what can we do with what we're learning? What all of this means for the consumer of this information is what they talk about a little bit more about in the discussion. So it's saying here that this falls in line with what has been shown in the research, in that iron overload causes a systemic metabolic remodeling that compensates for both an increase in oxidative stress as well as a decrease in glucose metabolism. So dietary iron overload increased the liver 2,3-hydroxybutyric acid, and its excretion, so it increases those levels. This is all indicative of the body trying to create more glutathione to neutralize the oxidative stress. It's also been used as that 2,3-hydroxybutyric acid has been used as a early biomarker for insulin resistance. So this is something that is going to be naturally high in people in pre-diabetic state, type 2 diabetes, type 1 diabetes as well. Now, to piggyback off of that, they say animals receiving the high iron supplement showed higher glucose levels. Okay, that makes sense, right? Because glucose metabolism is being changed. All of the byproducts of glucose metabolism are going down, and all of the other forms of energy being used, like fatty acids, like AMP and ADP, those are all increasing. Now, these authors don't go too much into detail in terms of that altered glucose metabolism and the increase in glucose. They suggest two possible explanations. Firstly, that iron loading might act as an activator of the Krebs cycle, which is fueled by substrates other than glucose. Again, we talked about that altered glucose metabolism where it's not using glucose fully. It is using something else to create energy in the body, which may be a result of decreased beta cell action. Again, they don't talk anything about beta cells here, but it has to put the bug in your ear about looking for ways that the body is compensating for all of this high stress and decreased glucose usage. And secondly, the paper mentions a lot about carnitine levels. So if you remember, we'll scroll back up to that first table talking about the changes in the blood levels of particular metabolites. When you start to look at the names of all the things that are decreasing, you can tell by the arrow on that right-hand side, there are a lot of things associated with carnitine. So L-acetyl-carnitine, stearoyl carnitine, propionyl carnitine, carnitine. There's a lot going on with carnitine. Now, what even is carnitine? You know, how can we address that? Is it a byproduct of a defense mechanism? Is it a deficient nutrient that is needed to address the oxidation going on? I'm not entirely sure of that yet, but this paper is acknowledging that in the iron overloaded condition, carnitine, you know, the low levels of carnitine, it's being consumed in the body so that there's not a lot going on in the blood. And, you know, it's up to us to see what that's all about. So you have to wonder to yourself, is carnitine something that can be employed as an antioxidant to fight oxidative stress? Or is it some kind of byproduct of that glucose imbalance in the body? And in the paper's conclusion, they go on to say that there is quite clearly a specific blood signature going on in this secondary iron overloaded state. An important thing to remember, this is not hereditary hemochromatosis. This is strictly iron overloading induced by diet alone. So in the 2 weeks of this high iron diet, something is happening at a systemic level that is affecting the degree of oxidative stress in the body, as well as the body's ability to or preference for using sugar, using glucose in the body. And it goes on to suggest that in this metabolic restructuring to compensate for the increased oxidative stress, we need to learn more about ways to address the iron imbalance and to return to a more natural.
So how cool is that? You're getting a live feed of the metabolic byproducts in these animals being fed this high iron diet. You're seeing increases in oxidative stress markers. You're seeing increases in glucose, as well as decreases in the byproducts of proper glucose metabolism. Now, for those of us with diabetes reading this research and thinking, okay, well, what am I supposed to do with this information? What I've done is I've looked at each of the trends in the metabolites, the increase in carnitine, the decrease in glutathione, the increase in like all of the byproducts of both managing oxidative stress as well as properly managing glucose levels. And what are things that we can supplement? What are things that we can eat that are going to positively impact those levels to help bring them back to what was being shown in the metabolic outputs of the normal chow. So for me, things that I'm paying attention to in this paper is I'm going to look into carnitine a little bit more. That is something that you can supplement. It's something that's fairly abundant in a whole foods diet. I want to know more about its ability as an antioxidant, as well as its place in the whole spectrum of proper glucose metabolism, as well as altered glucose metabolism, to see if it's something you want more of or something you want less of. But clearly there are changes happening in the iron overloaded state, as well as natural antioxidants. So things like glutathione, anything that you can take to neutralize reactive oxygen species, right? Because all of the, like the 2,3-hydroxybutyric acid, the methionine sulfoxate, those are all a result of the body trying to manage increased ROS, right? The more ROS you have, the more inflammation you're experiencing. So if there's something that you can take, there is something you can eat, or in your diet, something that you can take away that is going to prevent increased reactive oxygen species, that's something that I see as being effective in addressing the changes that we're seeing. So I know that is a lot of information, and I don't expect everyone to get that the first time through. I am going to link the paper in the description. You'll be able to see that, as well as a bunch of other of my resources regarding managing diabetes, things to supplement, things to eat, and go through it a couple times. Take the papers referenced in this specific paper and branch off into new areas of research. You're going to learn a lot about iron homeostasis. You're going to learn a lot about iron dysregulation in the states of disease. That's all something that you need to have a foundational understanding in to see how important the changes that are happening are. Okay? So for the time being, thank you so much for being here. I really appreciate it. This is the review for the metabolic signatures of dietary iron overload in the mouse model. I'm your host, Bowie. I hope you enjoyed.